notes with CBME based question answers, MCQ, and full zone for self-assessment. Then MCQs are also being provided from beginner level to expert level that consist of standard multiple choice questions. Then student can also book a slot for personalized counseling session in which there will be a direct interaction of the student with the faculty. Then we provide some subscription plans also, starting from basic to premium. So basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months. So the premium plan, the services include like pre-recorded classes, live class, live dot clarification, one-to-one personalized guidance, notes, quick revision examination module, and it costs around 50,000. So I would recommend all the students to take the premium plan of 12 months. And also for instant 20% discount, you can use my code, that is Aanchal 20. So on these days, we are going to have the session over jaundice, WBC, and platelets. So today we are having the class over RBC. So before proceeding, let's discuss a clinical case scenario that is of a 45-year-old male present to the emergency department complaining of fatigue, shortness of breath, palpitation for the past month. He denies any recent illness or significant medical history. So in examination, he appeared pale with conjunctival pallor noted. Laboratory investigation revealed a hemoglobin level of 8.2 gram per deciliter and hemocrit of 25% and mean cardiovascular volume is 76 FL and reticulocyte count of 1.5%. Further investigation revealed low serum iron level and elevated total binding capacity. The peripheral blood smear demonstrate a microcytic and hypochromic red blood cells, and which of the following is the most likely underlying cause of the patient anemia? So we have the option first, vitamin B12 deficiency, second, folate deficiency, third, iron deficiency, and fourth, hemolytic anemia, and the last is aplastic anemia. So again, we're going to discuss this clinical case scenario after understanding this topic. So starting from the introduction part. So the solids of the blood contain RBCs, white blood cells, and platelets. So the RBC delivered oxygen from our lungs to the tissues and organ, and white blood cells are responsible for the fight against the any kind of reflection, and also they are an important part of our immune system, right? Whereas the platelets are very helpful for that. They help in blood clotting whenever any person have a cut or wound. So this topic is of invariable interest and forms the basic of systemic physiology that we're going to discuss today. So as we can see in blood, various types of cells are there like WBC, platelets, RBC. So today we're going to discuss about the RBC in detail. So the function of blood is to deliver oxygen, remove metabolic waste, maintain temperature, pH, fluid volume, protection from blood loss also through platelets, prevent infection with the help of antibodies and WBC, and also blood is important for the transport of various hormones and some enzymes. Then there are some components they have shown in the slide that can be separated through the centrifuge machine, and as we can see here, the top coat is the plasma, and this buffy coat contains some WBC and platelets, and the bottom portion is here showing the RBCs. Then talking about the components of normal adult blood, so we have 8% whole blood and 92% other fluids and tissue, right? So we have 55% plasma and 45% formed element. So as we have discussed in the last class also about plasma. So in plasma, 7% is protein part, 91.5% water part, and 1.5% are solutes. And in protein, we have learned albumin, globulin, fibrinogen, and other also. And other solids, we have learned about electrolytes, nutrients, gases, regulatory substances, vitamins, and waste products. So in form elements, we have platelets, WBC and RBC. In WBCs also we have a classification as granulocytes and agranulocytes, in which we have neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So the in abnormal hemocrits, we have polycythemia. So polycythemia means increase in RBC production, above the normal range, right? As we have a normal range that differs from male to female. So if the RBC count exceeds above then the normal range, so that condition is known as polycythemia. And physiological polycythemia, there will be increase in RBC production due to hypoxic tissue, like what occur at high altitude. So whenever we do the CBC of the people who are living at the high altitude, so we always see the condition of polycythemia, means there will be increase in the RBC rate of those people who are residing at high altitude, right? So in blood plasma components, we have 50%. From 50%, we have 90% water, 8% solutes, proteins like albumin, alpha and beta globulin, gamma globulin, fibrinogen, and gas, right? And some electrolytes are also there. In Buffy coat, we found WBCs and platelets. Now, formation of blood cells. So formation of blood cells known as hemopoiesis, right? So as the name refers, hemo means blood cells and poiesis means formation. So this word hemopoiesis means the formation of blood cells. Okay. So for the formation of blood cells, we have two theories. The first theory is monophyletic theory, and the second theory is polyphyletic theory. Okay, so both the theories are different, and they are also stating some different concepts. Okay, so as the name mono means single, monophyletic means this theory states that all the cells are derived from the single mother cell. As we can see in the picture also, that all the different types of cells, like whether it is WBC, platelets, monocytes, neutrophil, B lymphocytes, T lymphocytes, all the different types of cells are derived from the single mother cells, from the single pluripotent stem cells. Now, what about the polyphyletic theory? So this theory states that separate stem cells are present in the bone marrow for each variety of blood cells. Okay, means this theory is stating that for RBC, different stem cell is there in bone marrow. For platelets, different stem cell is there. For WBC, different stem cells are there. Okay, but right now, what the theory we considered, that is the monophyletic theory, which is indicating that only single mother cell are present for the production of all the various type of cells. Okay, and that is known as pluripotent stem cells. Okay, so here, as we can see in the diagram also, this pluripotent stem cell is responsible for producing various type of cells. For example, from pluripotent stem cell, there will be arrival of myeloid stem cell. Then there will be formation of colony forming units of erythrocytes, from this to pro-erythroblast, from this to reticulocyte, and finally there will be formation of RBC. So that all these steps we're going to learn in detail in today's class about the formation of RBC, and that process of formation of RBC is known as thrombopoiesis, and that known as erythropoiesis. And for this erythropoiesis, we need a very important hormone, that is erythropoietin, that is secreted by kidney and liver both. Okay, and another thing is there that other various cells are also, we can see, like platelets, monocytes, different types of WBC, neutrophils, eosinophils, basophils, B lymphocyte, T lymphocyte. So about the formation of all these cells, we're going to discuss, and also we're going to learn in the other class. Like in platelet class, we're going to learn about the thrombopoiesis. In WBC class, we're going to learn about the leukopoiesis. So the formation, all this formation we're going to learn in different class, but in today's class we're going to learn about the formation of RBCs only, and that is erythropoiesis. Okay, so formed element of the blood, we have erythrocytes, leukocytes, and platelets, right? Okay, now starting with the erythrocytes. So this erythrocyte is a circular biconcave non-nucleated disc. Okay, so please remember all the student, then this RBC is always non-nucleated. Right? And why this is non-nucleated? So that it can give more space for hemoglobin, as hemoglobin is very important pigment because it is responsible for carrying oxygen, right? So this is anucleated, so can't reproduce, and repro in red bone marrow occurs, right? Now, what is hematopoiesis? Hematopoiesis means the production of RBC. The function of the erythrocytes to transport respiratory gas there. Then talking about the structure of the hemoglobin. So as we know, it contains 2 alpha and 2 beta chain, and erythrocytes lack mitochondria always. Now, one RBC contains 280 million hemoglobin molecule. One RBC is responsible for carrying 280 million hemoglobin molecules. And in male, the RBC, the normal RBC count is 5 million. And in women, it is 4.5 million cells. Then talking about the lifespan of the RBC, so the lifespan is 100 to 120 days. And after this lifespan, all the RBC gets destroyed inside spleen. That is also known as graveyard of RBC. Okay. Okay. Now, another thing is this, that it is always biconcave in shape. Okay. Now, what is the advantage of this biconcave shape? The advantage is that it allows considerable change in cell volume, so that it can withstand considerable change of osmotic pressure and also resist hemolysis. Okay. One advantage is this. Second, it allows easy folding of RBC on itself when it passes through the capillary. Okay, so we have done 2 advantages. Then the last one is that hemoglobin remain distributed in the center of RBC, which facilitates optimum and quick exchange of gases like oxygen and carbon dioxide. Okay, so this is all about the shape. Why the shape of erythrocyte is biconcave. Okay, now talking about the composition of this. So whenever we look at the composition, so okay, so 62.5% we have water content. Then 35% we have hemoglobin content, and rest 2.5% we have other substances also. Okay, so this is all about the composition of erythrocyte. Okay, so as we have already discussed that the count what is a normal count at in female and what is a normal count in male, and one thing is also remaining, that what is the normal count at birth. So at the time of birth, the count of RBC will be 6 to 7 million per microliter. Okay, so this will be the count of RBC at the time of birth. Now, hematopoiesis or hemopoiesis. So this is for the blood. This terminology is used for the blood cell formation, and as we know, this occur in red bone marrow of axial skeletal girdles and proximal epiphyses of the humerus and femur. Okay, so these are the site of formation of blood cells. Then hemocytoblast is the hemopoietic stem cell, which give rise to all the formed elements, as we have seen in this picture also, that this mother cell is responsible for giving rise to all the blood cells. Okay, so this is known as hemopoietic stem cell. and sometimes it is in some book they have given pluripotent stem cells also. It gave rise to all the formed elements, hormone and growth factor push the cell towards a specific pathway of blood cell development, and new blood cell enter the blood sinusoids. Now, erythropoiesis. Erythropoiesis means the formation of red blood cells or red blood cells production. So, a hemocytoblast is transformed into proerythroblast, and proerythroblasts develop into early erythroblasts, and from early erythroblasts, there will be development of reticulous normocytes, then normocytes to reticulocytes, and finally erythrocytes. Now, the sites of erythropoiesis. So, talking about the site of erythropoiesis before birth means during intrauterine life. So, during intrauterine life, the formation of RBC will take place at yolk sac, liver, and spleen. After birth, normally large cells of bone marrow of certain bones like vertebra, sternum, hip, and of long bones, flat bones like ribs, pelvis, skulls are responsible for the formation of blood cells. And spleen is sometimes also the site of formation that is secondary to some specific kind of disease. Then talking about the erythropoiesis, okay. So what is erythropoiesis, and what are the different types of stages in which the development of RBC will take place, that we're going to understand, okay. So first we're going to understand the erythropoiesis in intrauterine life. So erythropoiesis in intrauterine life. So erythropoiesis in intrauterine life, we have 3 stages. First, we have mesoblastic stage. Second, we have hepatic stage. Then third, we have myeloid stage. So in mesoblastic stage, in early embryo up to 3 months. Okay, up to 3 months. The formation of RBC will form from yolk sac. Okay, so here in first stage of intrauterine life, the RBC formation will take place in yolk sac. Okay. Then in hepatic stage, as the name refers, hepatic means liver. Okay. So this stage comes after 3 months. Okay. And before 6 months. And here the site of formation of RBC will be liver and spleen. Okay. Then finally, the last we have myeloid stage. So here this myeloid stage will occur between 6 to 9 months of fetal life, and in myeloid stage, the site of formation of RBC will be bone. Okay, so this is all about the erythropoiesis which occur during intrauterine life. Okay, now we're going to discuss the characteristic feature of erythropoiesis. Okay, so the characteristic features we have. First, that there is reduction in cell size. There is a reduction in cell size. Second, the cytoplasm increases in amount and nucleus decreases. Okay, so these are the characteristics feature of the erythropoiesis. Okay, during adult life. Right now we are discussing. Now we're going to discuss all the stages. Okay, and this is going to be very important. This slide is going to be very important. So first we're going to discuss about the various stages that occurs during erythropoiesis. So starting from the hemocytoblast, that is also known as stem cell, right? Then second we have proerythroblast, then early normoblast, then we have intermediate normoblast, then we have late normoblast. In late normoblast also we have two stages, early and second late. Then finally we have reticulocyte, and finally the last is erythrocyte. So we're going to discuss all these stages in comparison with cell size, then nucleus, then cytoplasm, and finally division, means mitosis. Okay. So starting from the cell size, so as we know, the size of the mother cell is always larger, and the size of the mature cell, means the size of the erythrocyte, will be lesser. Okay, so as the stage will proceed, means the first stage will be hemocytoblast, then proerythroblast, then early normoblast, then intermediate normoblast, then later normoblast, then reticulocyte and erythrocyte. So as the stages will proceed, the size of the RBC will decrease, right? The size will decrease. So the size of the hemocytoblast will be between 19 to 23 μm, and the size of the mature erythrocyte is 7 to 8 μm. Okay, then the size of the nucleus is also decrease, right? So size of the nucleus decrease because in mother cell, means in the stem cell, the nucleus will be present, but as we have discussed, in erythrocyte the nucleus is not present. So as the stages will proceed, the size of the nucleus will also proceed, and one stage will come where finally the nucleus disappear. Okay, so what is that stage where nucleus disappear finally? So that is late normoblast. Okay, disappeared. Okay, and in reticulocyte, no nucleus is there, only some remnants of RNA present. Okay, and the size will decrease till this stage. Okay, now what about the cytoplasm? As the size of the nucleus will decrease, so size of the cytoplasm will increase. Okay, so the size is going to increase. I can write like this. And one thing also happen, that there will be sudden appearance of hemoglobin. So as we know, in mother cell, in stem cell, hemoglobin is not present, but there will be sudden appearance of hemoglobin at which stage? At intermediate normoblast. Okay, so hemoglobin appear at intermediate normoblast. Okay, now talking about the division, so as we know, RBC doesn't have nucleus, so where there is no nucleus, there will be no division, right? So division will be present till the intermediate normoblast, but as the nucleus will get disappeared finally in late normoblast, the mitosis stop here. Okay, otherwise it is present in all the stages. Okay, so this is all about the stages of erythropoiesis, and this is a very important slide. If you get the question over the stages of erythropoiesis or over the erythropoiesis, so you need to draw this table, right? Okay. Now we're going to discuss all these stages we have discussed, right? The first was hemocytoblast, then proerythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte, and erythrocyte. So this is the mature one, right? And here also we have discovered that in all these stages, the size of the nucleus will decrease and finally disappear in the late normoblast, and there will be sudden appearance of hemoglobin in intermediate normoblast. Okay, so this is all about the erythropoiesis. Now let's discuss about the another important topic, that is regulation. Okay, so for regulation, we have two things. The first, we have general factor, in which we're going to learn about hypoxia. Another thing we have spatial maturation factor. In spatial maturation factor, we're going to learn about dietary factor, Castle intrinsic factor, and extrinsic factor. All these factors we're going to learn and we're going to discuss one by one. Okay, so first we have this hypoxia. Okay, now what is hypoxia? Hypoxia means the deficiency of oxygen in the at the tissue level, right? So hypoxia is the most potent stimulus for the production of RBC, right? Hypoxia will stimulate bone marrow for the production of RBC, right? How? As the person visited to high altitude, right? As the person will visited to high altitude, there will be deficiency of oxygen because there is a low partial pressure of oxygen at high altitude. Okay, so at that time, the person will suffer this kind of thing. Okay, the person will have the hypoxic condition. So this hypoxia will stimulate our organ like liver and kidney. Okay, now kidney will release erythropoietin and liver is going to release erythropoietinogen. That is a kind of alpha globulin. Okay, and that is also responsible for the release of erythropoietin. Now this erythropoietin is going to stimulate bone marrow and there will be increased production of RBC. Okay, so in this way, the hypoxia will be responsible for increasing RBC production with the help of the hormone, that is erythropoietin, secreted by liver and kidney. The erythropoietin is going to stimulate bone marrow and there will be increase in the production of RBC, right? Then another thing we have this spatial maturation factor that we're going to discuss one by one. So first we have dietary factor, right? So in dietary factor, protein enable globin formation. Also some other thing like iron, magnesium, copper, cobalt are responsible for heme formation, right? Then another thing, calcium increases the iron absorption from GIT. And the last, that vitamin C, vitamin B12 and folic acid are useful in the synthesis of nucleic acid. Okay, so this is all about the dietary factor. Then another thing we have Cassell's intrinsic factor. Okay, so this is a kind of glycoprotein, right? And this is produced by parietal cells of stomach. Okay, now this how this Cassell's intrinsic factor is useful because it binds with vitamin B12, and whenever this vitamin B12 reaches in the terminal portion of ileum, so at that time this helps in the absorption of vitamin B12. Okay, so and the vitamin B12 is very important for the maturation of RBC that you have to learn. Okay, then another thing we have extrinsic factor. So this extrinsic factor is present in certain foods and essentially in vitamin B12 and folic acid, and also they play an important role in the synthesis of DNA. Okay, and this intrinsic factor, this extrinsic factor with intrinsic factor is known as hematinic principle, which is useful for the maturation of RBC, right? Okay, then another thing we have in regulation, too few RBCs leads to tissue hypoxia, as we have seen what happens at high altitude. So too many RBC increases blood viscosity. Blood balance between RBC production and destruction depend upon various hormone control, for example, erythropoietin, and adequate supply of iron, amino acids, and vitamin B, various vitamins. Okay, not B. It should be B12. Okay, so all these are responsible for the regulation of erythropoiesis. Okay, then we have erythropoietin. As we have seen, there will be direct stimulation for erythropoiesis, which is released by kidney in response to hypoxia. Then causes of hypoxia, hemorrhagic condition or increased RBC destruction reduces RBC number, and if there is insufficient availability of hemoglobin in case of iron deficiency, that time also. And if there is reduced availability of oxygen at high altitude, then at time also there will be condition of hypoxia will arise. Then what are the effects of erythropoietin? Okay, so more rapid maturation of committed bone marrow cells, and there will be increase in circulating reticulocyte count in 1 to 2 days. Testosterone also help enhance erythropoietin production, resulting in higher RBC count in males. Okay, now what happened there when there will be formation and destruction of RBC. Okay, so now in one slide we're going to learn that how there will be formation of RBC will occur and destruction of RBC will occur. Okay, so what happened, as we have seen, there is formation, erythropoiesis occur in red bone marrow. Okay, so RBC will form in red bone marrow with the help of iron, globin, vitamin B12, and the hormone erythropoietin. Okay, so various components are very important that already we have seen. So after 120 days, means when the lifespan of RBC will end, so what will happen? This RBC will get burst out. Okay, and suddenly that hemoglobin will get ejected out from the RBC. Okay, so RBC cell death and phagocytosis will occur, that occurs in spleen, liver, or bone marrow. So what will happen? From heme, there will be formation of biliverdin, that already we have discussed in hemoglobin class, that how there will be formation of biliverdin will occur from the hemoglobin, and also this globin will enter into amino acid pool cycle and reuse for protein synthesis, and this biliverdin get converted into bilirubin and get stored and get transferred into liver for the conjugation, and from conjugation, whenever the food will come in the intestine, so it will get entered into the small intestine for fat emulsification, and from here there will be formation of stercobilinogen and urobilinogen in kidney. And this stercobilinogen is going to excrete out from feces, and urobilinogen is going to excrete out from urine. Right, now this iron also is there. Okay, so iron get converted into ferritin and finally get stored inside liver, and whenever there is requirement of this ferritin for the formation for the synthesis of hemoglobin, again it get transferred into bone marrow for the formation of various blood cells. Okay, so this is all about the formation and destruction of RBC that we have discussed in this one slide. Okay, so now we have seen the pluripotent stem cell. From this, there will be formation of myeloid stem cell and various type of cells like erythrocyte, platelets, and various WBC will form. And from this pluripotent stem cell, there will be formation of lymphoid stem cell and B cells and T cells will form. Okay, now we have a MCQ and all the student will have 10 seconds to answer that in fetal embryonic life, the RBC cells are formed in. The first option is spleen and liver, liver and kidney, kidney and lungs, and pancreas and liver. So I know all the student knows the answer that already we have discussed, right? Yes, the answer is spleen and liver. Okay, the answer is A, spleen and liver. Okay, now another question we have. All are required to stimulate erythropoiesis except estrogen, erythropoietin, testosterone, and thyroxine. Again, you have 10 seconds to answer. Yes, the answer is estrogen. All are required. Erythropoietin is required, testosterone is required, thyroxine is required for erythropoiesis, but estrogen doesn't have as such any significant role for erythropoiesis. Okay, now we're going to discuss anemia. Okay, so what is anemia? This is a clinical condition in which oxygen carrying capacity of blood get reduced. Okay, and this is characterized by reduction in RBC, right? And there are some grading of anemia like mild anemia, moderate anemia, and severe anemia. Okay, so as we know, the normal level of hemoglobin is between 12 to 16. Okay, now whenever the level falls below 12 and above than 8, so that condition is mild anemia. Whenever the level of hemoglobin falls below 8 but above 5, that is moderate anemia. And whenever the hemoglobin level goes below 5, so that is severe anemia, in which the transfusion of blood will be needed for the patient. Okay, so anemia when blood flow has low oxygen carrying capacity, insufficient RBC or iron deficiency. So factors that cause anemia like exercise, vitamin B12 deficiency, and polycythemia is a condition in which there will be excess of production of erythrocytes and increase in the viscosity of blood cells, and the count of the RBC will range from 8 to 11 million cells. Right now, usually caused by cancer, tissue hypoxia due to dehydration, naturally occur at high altitude, and also there is a condition called blood dropping, in which the blood there will be the blood will remove in 2 days before event and then replaced to the person. In usually that occurs in the cases in athletes usually. Okay, then another thing we have sickle cell anemia. So that is results from a change in just one of the 287 amino acids in the beta chain. Okay, so the mutation will take place in beta chain and found in 1 out of 400 African Americans. Abnormal hemoglobin crystallize when O2 content of blood is low and causing RBCs to become sickle shaped. And homozygous for sickle cell is deadly, but in malaria infested countries, the heterozygous condition is beneficial. Okay. Now, finally, we have the summary. So, blood is composed of cells, cell fragment, and an aqueous solution. Blood makes up about 8% of human body weight. It contains erythrocytes, leukocytes, thrombocytes, and plasma. Now, maintenance of osmotic pressure, transport of fatty acids, bilirubin, bile acids, steroids, hormone, pharmaceutical drugs, inorganic ions, globulin participate in the immune system, that is immunoglobulins, and also act as transport protein, and fibrinogen are involved in clotting process. Okay, so this is all about the this RBC, in which we have learned about the shape of the RBC, we have learned about the function of the RBC, then we have done the hemopoiesis, the theory stating hemopoiesis, then we have learned erythropoiesis stages during intrauterine life, and we have done the erythropoiesis during adult life. Okay, now one more thing is there, that also all the students should learn about the difference between red bone marrow and yellow bone marrow. Okay, so this red bone marrow consists of numerous blood cells and all kind of their precursors, right? Now, this yellow bone marrow consists of fat cells, blood vessels, and a minimal framework of reticulum cells and fibers. Second thing, this is known as active marrow because it is responsible for the production of all blood cells, and this is inactive marrow. Okay, it includes end of long bones, right? And it includes shaft of bones. Okay, so this is all about the major difference between red bone marrow and yellow bone marrow that all the students should learn. Okay, so this is all about today's class, and this is all about the about the topic of RBC. Then also we have a question, clinical case scenario. Again, we're going to discuss, right? So we have learned that in further investigation reveals low serum iron levels and elevated total iron binding capacity, right? And the peripheral blood smear demonstrate microcytic and hypochromic red blood cells. So it is clearly indicating that there is a condition has arisen, that is of anemia, and the low serum iron levels indicating that there is iron deficiency. Okay, so that is the answer will be iron deficiency. Okay, so I hope all the students find this class useful. So thank you so much for today's class. Thank you so much for watching my class.